Good day everyone, this is Bonroel Gaspina and now we are down to the last part of Group 2's report. Concept number 44.5, Hormonal circuits link kidney function, water balance, and blood pressure. Okay, I believe by this point we all know what hormones are and how important their function is. Hormones control hundreds of reactions in our body. They are signaling molecules that tells our body when and how to behave. Hormone circuits may consist of your glands, tissues, or organs that produces and regulates hormones in your body. When these hormone circuit is out of balance, many ugly symptoms may arise and could cause health problems. Each of these components acts as a hormone cascade that begins with a control center in the brain. Now, hormone cascade is when some hormones causes other tissues or organs to release hormones. Okay, all of the things that I just said was just a foreshadowing of my report. Now let's proceed to the main topic. In mammals, both the volume and osmolarity of the urine are adjusted according to an animal's water and salt balance and its rate of urea production. Osmolarity refers to the number of particles of solute per liter of solution, and urine osmolarity measures the concentration of particles in urine. While urea is a waste product that travels through your blood to the kidneys, which then filters it out of the blood. It is then carried out of your body through urine. Now, in situation of, let's say for example, if a mammal has a very high salt intake and a low water availability in its body, a mammal can excrete urea and salt in small volume of hyperosmotic urine with minimal water loss. Now, in the opposite situation, if a mammal has a very low salt intake and a high water intake or availability in its body, the kidney can instead get rid of the excess water with little salt loss by producing large volume of hypoosmotic urine. To further elucidate, let's have an example. The South American vampire bat illustrates the versatility of the mammalian kidney. But how? Vampire bats are nocturnal animals, meaning they are most active at night, and they seek for prey at night. As the name suggests, they feed on blood from other animals. So, at nighttime, they fly for hours on end to find food. Now, imagine you in IIT campus at 12 noon craving for donuts. However, there are no donuts in IIT or in Tibanga, but you really just want donuts for lunch. Lunch? That's all you can think of. So you thought, oh, there are donuts stationed near the cathedral. However, as you walked out of campus, you can't find any vacant jeepney, so you had no choice but to walk on foot. That's how dedicated you are to find or eat donuts. Now, you are near the donut station and you are tired and exhausted from all that walking, and upon arriving to your destination, you ate only 2 donuts. Is that enough to replace the energy you've lost? Does 2 donuts suffice? No. Of course, you eat 2 boxes of donuts to satisfy your cravings. Now, similarly, let's go back. The South American vampire bat fly long distances for hours to find prey, and when they do, they drink and consume blood as much as possible, so much that it almost equals the size or the mass of their own body. This will result to them not being able to fly, but to counter or to solve this problem, the vampire bat excretes dilute urine either while drinking blood or 2 minutes after consumption of blood. What happened here is that the kidney offloads water absorbed from the blood and excretes large volume of dilute urine or hypoosmotic urine. Having lost enough water weight, they can then fly back to their roosts or cave. In the roost, vampire bats encounter a new regulatory problem. You see, most of the nutrition it derives from blood comes in the form of protein, and digesting protein generates large quantities of urea, which can cause health problems. But the vampire bat lacks the access to the drinking water necessary to dilute this particular substance. So, what it does is that their kidneys shift to producing a small quantity of highly concentrated urine or hyperosmotic urine. It is an adjustment that disposes of the urea load while conserving as much water as possible. The vampire bat's ability to alternate rapidly between producing large amounts of dilute urine and small amounts of very hyperosmotic urine is an essential part of its adaptation to an unusual food source. Okay, let me introduce to you the hormone that helps the kidney control the amount of water and salt in the body and the amount of urine that is made. This hormone is called the Antidiuretic Hormone, or ADH. ADH, also known as vasopressin, is a key hormone in this regulatory circuitry, and it is a combination of nervous and hormonal controls that manages the osmoregulatory function of the mammalian kidney. Osmoregulation, in simple terms, is the process of regulation of water and salt for an organism. Now, how does ADH work? Well, it all begins with a stimulus or a trigger. So, the preferred set point of blood osmolarity in our body is around 300 milliosmoles per liter. If the blood osmolarity increases more than 300 milliosmoles per liter, that means that your body needs water or that you are dehydrated. In the hypothalamus, there are osmoreceptors that detects this change in the blood osmolarity, which then tells your brain that you are thirsty. And by drinking water, your blood osmolarity decreases to a set point. Simultaneously, this stimulus will prompt the posterior pituitary gland to release ADH. By the way, the supraoptic nucleus synthesizes ADH and stores it in the posterior pituitary gland, which again is released due to high blood osmolarity. This will then travel to your kidney into the collecting duct, which eventually increases its permeability and reabsorb water in the body rather than being urinated out. This is to prevent further increase in blood osmolarity. This process is the work of your body and ADH in attempt to reach homeostasis. And the cycle repeats. But the question is, how does ADH make this happen? Let's take a closer look. This is a collecting duct cell. After the release of ADH, it will trigger an intercellular cascade inside the cell. It binds with vasopressin type 2 receptor, which activates the stimulatory G protein, which causes it to move along the membrane and activates an enzyme called AC, or adenylate cyclase. AC takes ATP from the cell and converts it into CAMP, or cyclic adenosine monophosphate, also called as the second messenger signaling molecule. This cyclic AMP generates a very important protein called pKa, or protein kinase A, which regulates a lot of activities inside the cell. pKa stimulates genes in the nucleus and makes specific proteins that eventually emerges in vesicles. These proteins are then called aquaporin 2. pKa phosphorylates these vesicles containing aquaporin 2 and modulates exocytosis, which is a process that fuses the aquaporin 2 containing vesicles to the membrane. Aquaporin 2 has a major role in this procedure because they are water channels or water selective tubes that increases the permeability of the cell. So what it essentially does is that it takes water from the collecting duct and back to the bloodstream or into the body. Otherwise, it will get urinated out. This explains why you excrete reduced urine volume and hyperosmotic urine. As the osmolarity of the blood subsides, a negative feedback mechanism reduces the activity of osmoreceptor cells in the hypothalamus and ADH secretion is reduced. But what if there is a mutation that prevents ADH production, or when ADH receptors are blocked, thus lack ADH response? This can cause severe dehydration and solute imbalance due to the production of urine that is abnormally large in volume and very dilute. This condition is called polyuria, and a person can also develop polydipsia or excessive thirst. These symptoms are the conditions of diabetes insipidus, from the Greek word to pass through. Aquaporin 2 mutations causes the aquaporin 2 proteins to be misfolded into an incorrect three-dimensional shape. This misfolded protein is trapped within the cell, where it is unable to reach the cell membrane to transport water molecules. This relates to the condition diabetes insipidus. On the other hand, if there is no aquaporin mutation and if ADH secretion is high, then a mammal can develop SIADH, or Syndrome of Inappropriate ADH Secretion. SIADH causes the body to retain too much water and could lead to pulmonary diseases. And by the way, even without genetic changes, certain substances can alter the regulation of blood osmolarity. For example, an alcoholic drink can disturb water balance by inhibiting ADH. Alcohol functions as a diuretic means, and it makes you urinate more. Therefore, you would be low on water. Therefore, you would be dehydrated. A diuretic or diuretic is any substance that promotes diuresis, the increased production of urine. And vasopressins are supposed to oppose this because they are an antidiuretic hormone. All in all, blood osmolarity, ADH release, and water reabsorption in the kidney are all linked in a feedback loop that contributes to homeostasis. The secondary regulatory mechanism that has maintained homeostasis by acting on the kidney is the Renin-Angiotensin-Aldosterone System, or RAS. It is a system that gets activated when blood volume and blood pressure drops, and these three are released in that particular order. Let us first talk about renin. The filtration system of the kidney is called the nephron, and there is about 1 million nephron in each kidney. If we take a single nephron and zoom it, this is what we will see. The juxtaglomerular apparatus right here is responsible for releasing renin. It is a specialized structure formed by the distal convoluted tubule and the glomerular afferent arteriole. Around the afferent arteriole, there are juxtaglomerular cells, and these cells are actually baroreceptors, meaning they detect pressure. So when blood passes through this arteriole, it detects blood pressure and volume. Low blood volume triggers the juxtaglomerular cells to release renin enzymes. The blood goes through here in the glomerulus and gets filtered. The filtrate is gonna move slowly because of the low blood pressure, and this could result to the useful substances such as sodium and water to get pulled out or eliminated. The chemoreceptors in the muscular densa cells in the distal convoluted tubule detects this insufficient amount of sodium and communicates with the juxtaglomerular cells to also release renin. After the release of renin, it goes into the bloodstream and comes across angiotensinogen from the liver and converts it into angiotensin 1. Angiotensin 1 inevitably will travel to the lungs where angiotensin converting enzyme or ACE are dominant, or perhaps in the vascular endothelium of other tissues where ACE is present. ACE converts angiotensin 1 into a peptide called angiotensin 2. So what it does is that it functions as a hormone and is a generalized vasoconstrictor, and it mainly constricts arterioles resulting to a higher blood pressure. The second thing it does is that it goes to the smooth muscles around the afferent arteriole and constricts it, leading to the increase in filtration rate and increase the number of sodium in the distal convoluted tubule. Angiotensin 2 also travels all the way to the cortex of the adrenal gland and stimulates it to release aldosterone. Aldosterone travels to the distal convoluted tubule and tells it to take sodium and reabsorb it back to the body, because sodium promotes water retention. Wherever sodium goes, water follows. In short, an increase in sodium and water reabsorption will result to an increase in blood volume, therefore increase in blood pressure. That is because blood pressure is directly proportional to the blood volume. The RAS operates as a part of a complex feedback circuit that results in homeostasis. A drop in blood volume and pressure triggers renin release from the juxtaglomerular apparatus. In turn, the rise in blood pressure and volume resulting from the various actions of angiotensin 2 and aldosterone reduces the release of renin. The function of ADH and RAS may seem redundant, but this is not the case. Both increase water reabsorption, but they counter different osmoregulatory problems. ADH responds to an increase in blood osmolarity. However, a situation that causes an excessive loss of both salt and body fluids, a major wound for example, will reduce blood volume without increasing osmolarity. Hence, this will not affect ADH release. But RAS will respond to the drop in blood volume and pressure by, again, increasing reabsorption of sodium and water. Thus, ADH and RAS are partners in homeostasis. There is, though, a hormone that opposes RAS. This hormone is called the atrial natriuretic peptide. Let's just call this ANP. This particular hormone is secreted from the atria of the heart in response to an elevated blood pressure. ANP has a few functions. First, it inhibits the release of renin from the juxtaglomerular apparatus. Second, it inhibits salt reabsorption by the collecting ducts. And third, it reduces aldosterone release from the adrenal glands. These actions lower blood volume and pressure. Thus, the ADH, the RAS, and ANP provide an elaborate system of checks and balances that regulate the kidney's ability to control the osmolarity, salt concentration, volume, and pressure of the blood. In all animals, certain of the intricate physiological machines we call organs work continuously in maintaining solute and water balance and excreting nitrogenous wastes. The details that we have reviewed in this chapter only hint at the great complexity of the neural and hormonal mechanisms involved in regulating these homeostatic processes. And that concludes our report. Thank you so much for listening.